So we decided to test osmosis in two different ways. To start, we did a lab with a phenolphthalein gel, and so what happened is we had the phenolphthalein and it was pink to start, and then we added vinegar and acid onto it, and when you mix an acid and phenolphthalein, it turns clear. And so what we actually were able to do is observe the color change throughout like 25 minutes. And our cell, aka the gel, was shaped like a penguin, and so as you can imagine, thinking of a penguin, there's lots of curves, and so this would increase the surface area, which we hoped would increase the rate of diffusion and like how fast the vinegar would come in and turn the gel from a pink to a clear color. But the results of our experiment were actually a little different than we expected, because obviously at 0 minutes we had completely pink gel, no white, and then at 5 minutes it was mainly pink, but the outer edges had like started to turn white, and then 10 minutes that whitening progressed, and so there was a significant amount of white around the edges, and then at 15 minutes there was more white than pink in the gel now, so there was like that flip, and then at the ending 25 minutes, the majority of the cell, aka the gel, had lost its pink color, and only like the innermost part was pink, but it was still like a significant amount of pink, which we weren't expecting.
Yeah.
So our gel had it was really thick. Where we put our placement thing in, it was super thick, and so the experiment didn't go through all the way. So a large portion of the middle was remaining pink, and also the seahorse was a larger cutout, so it took a lot more time and a lot more surface area. So it didn't go through all the way.
So my group had the shark-shaped gelatin, which is the most festive in my opinion. We did 2 sharks to get like a base to see like what it would be like, as opposed to just having one gel piece. So when we did it, it took a while to kind of get going, but the increased surface area, because all the curves in the shark, I think helped speed up the reaction and help neutralize the phenolphthalein pretty quick. So it went from pink to clear pretty quickly for our group compared to others, because we had a lot of surface area showing.
We then decided to test the sucrose levels in potatoes.
So our hypothesis was that potatoes placed in a higher sucrose concentration, so those being like 1 molar, 0.8, 0.6, would lose more weight, and then those placed in a lower sucrose concentration, like the 0.2, 0.4, would lose less weight. And so the way we would match up the solutions and determine that our whole experiment would just be like by taking the before and after weights and comparing them and then matching them.
The materials needed for this lab are potatoes, 6 graduated cylinders, variations of sucrose solution, 1 molar, 0.8 molar, 0.6 molar, 0.4 molar, and 0.2 molar, distilled water, a balance, bore to push out the potato, a tool to push out the potato out of the bore, and a petri dish. The variables in this lab are as follows: the independent variable, sucrose solution concentration; the dependent variable, the weight of the potato; the constants, the initial size of potatoes, about 0.035 ounces, 50 milliliters of sucrose solution, and the duration of soak, overnight; the control, the potato soaked in regular water.
In this lab, we tried to match different sucrose concentrations to different colored beakers based on the weight of a potato chunk after it was in the solution overnight. 3 types of solutions in which osmosis occurs, and these are respectively hypertonic, which is what we're going to be discussing in this lab, hypotonic, and isotonic. So hypertonic occurs when there's a higher concentration of solute on the outside of the cell as compared to the inside, and this causes the cell to shrink. Hypotonic is when there's more solute on the inside rather than on the outside, and isotonic, that should be a T, isotonic is when there's like a balance on the outside and the inside of solute. This goes along with the principle of osmosis, which is essentially passive transport across the membrane of a cell. This means that the cell doesn't use energy to do this transport because it goes along with a concentration gradient. Concentration gradient means that molecules flow from high to low concentration. And this makes sense because you wouldn't use energy to flow from high to low since they're already seeming to burst at the seams and they're just waiting to get into a place that's has less of them, right? So if you're in a crowd, you're gonna want to move to somewhere that's not as crowded. That's just the natural thing to do. It doesn't require energy to push and shove people in the crowd, you know? So solution, there's a higher concentration of solute on the outside of the cell. So here's your cell, right? This is your cell. And in the cell, say you have 30% solute inside the cell, solute, and then on the outside you have 70% solute. So if you have 30% solute on the inside, that means you have to have 70% water. On the outside you have, outside you have 30% water. So there's a high concentration inside the cell, right? So this water is going to move out through the plasma membrane, causing the cell, as a result, to shrink up, right? So it wants to become even with the space outside. So this is a cell in hypertonic solution. So this is what we're studying in this lab because the sucrose has different concentrations of sugar, but it has a lower concentration of water on the outside than it does in a plant cell, in a potato cell in this instance. So we're going to measure the weight lost by each potato cell, say potato cell, in each solution, because the higher the concentration of solute, the higher concentration of solution, the higher the rate is of weight loss that's going to happen in each cell.
In order to complete this lab, here are the instructions that must be followed. Label the 6 graduated cylinders A through F. Using a potato, take a cork borer and extract 6 selections of potato of the same quantity. 3. Weigh each of these potato pieces on a balance. Petri dish is equal to 0.015 ounces. 4th, put each of the 6 potato pieces in a separate graduated cylinder. 5. Pour 50 milliliters of a different solution into the other 5 graduated cylinders. 6. Pour 50 milliliters of distilled water into the 6th graduated cylinder. 7. Observe the changes in weight of each potato after a day of soaking in the sucrose solution. In this lab, the goal was to determine if higher concentrations of sucrose solution will affect the potato's weight than lower concentrations. We hypothesized this because of the tendency of water to move along a concentration gradient. After an overnight soak, we decided to weigh the potato pieces from the unidentified sucrose solutions. Here were the results. Beaker C was the only solution that had the same final weight as the starting weight, even including our control variable potato weight.
The solution that we think had the same water potential as the potato was solution C because the potato placed in that solution did not gain or lose any weight, which means that the water potential would not move from the area of high to low or low to high. And then we think also that if the potato was placed in a dry area for several days before the experiment, the potato's weight would increase greatly because the water potential of the potato initially would be very low, and since water moves from areas of high to low potential, there would be a lot of water moving into the cell of the potato. We think that it could be possible to use the same experiment with an animal cell, but the results would probably be different because the structure of animal cells is different than that of plant cells. For example, plant cells have a cell wall, but we do think that it would be the same for fungal cells because fungal cells are similar to plant cells in structure. They both have a cell wall, so the experiment would probably be similar.
Okay, so drawing our conclusions, we determined that solution B had the highest concentration of sucrose, while concentration C had the lowest. We determined this by looking at the weight loss, and so solution B, the potato soaked in solution B, had a weight loss from 0.035 to 0.02, and so that was like our most dramatic weight loss, while solution C, the potato in that one, actually didn't change at all. And so that's how we drew those conclusions. We also determined that we had some trouble determining which solution was 0.6 and which one was 0.8. We determined that it was either A or E, but the water potentials and the weight loss were so close that it was hard to determine exactly which was which.
Okay, so the potential errors in our experiment could be that we did not weigh each potato individually. We just weighed one and assumed that they were all the same weight. So our calculations could have been off. And even though we used the same tool to make all of the potatoes, they might not have all been the same size. So that variable may have not been controlled and could have conflicted with the results of the experiment.